Let me begin by introducing our speaker. Our speaker is Professor Abba Gumel. Abba is a foundation professor at the School of Mathematics and Statistical Sciences of Arizona State University. He received his undergraduate degree in mathematics from Bayero University in Kano, Nigeria, and a PhD in mathematics from Brunel University in London. He was a full professor in the Department of Mathematics at the University of Manitoba in Canada before becoming a foundation professor of mathematics at Arizona State University in 2014. His research work focuses on the qualitative theory of nonlinear dynamical systems, with emphasis on the transmission dynamics and control of emerging and re-emerging infectious diseases. He has held various administrative positions, including the Deputy Director of the Mathematical and Computational Modeling Sciences Center at Arizona State University, the Director of the Institute of Industrial Mathematical Sciences, and Secretary of the Canadian Applied and Industrial Mathematical Society. Professor Gumel has written over 150 peer-reviewed publications, mentored numerous graduate students and postdoctoral fellows, and edited 3 books. He received numerous research awards and honors, including the African Mathematical Union Medal for Excellence in Applied Mathematics by an African under the age of 40, the RH Award for Interdisciplinary Sciences, which is the highest research award given to junior faculty at the University of Manitoba. He was elected Fellow of the African Academy of Sciences, the Nigerian Academy of Sciences, the ASU Santa Fe Institute Center for Biosocial Complex Systems, and the African Scientific Institute. It is both my pleasure and my honor to introduce Professor Gumel, who will speak on the mathematics of infectious diseases. Professor Gumel.
Thank you very much, Professor Weintraub, for the very generous introduction. Also very thankful to the American Mathematical Society for the immense honor to present this highly distinguished public lecture. But all credits must go to the many students, including high school, undergraduate, graduate students, postdocs, mentors, research collaborators that worked with me over the years from around the world. I mean, credits goes to them. And I'd also like to thank everyone on this line, on this Zoom link, for sparing their time to attend. This lecture I would like to dedicate to Professor Lee Lorch, major inspiration in my life and career, brilliant mathematician, former professor of mathematics at York University in Canada. For many reasons, Professor Lorch was a brilliant mathematician, a mentor, and an icon of equity, inclusion, and women empowerment and social justice in the world. And he received so many awards for his excellence in mathematics and also for his advocacy for racial justice. So he was a great advocate for education opportunities for women and underrepresented minority advocacy. His work on advocacy to end racial segregation actually cost him academic appointments in the 40s and the 50s, necessitating his self-exile to Canada in 1959. He studied at University of Alberta in Edmonton before moving to York University in North York, near Toronto in Canada. He was a fellow of the Royal Society of Canada, fellow of the American Association for Advancement of Science. He received Lifetime Achievement Award by National Association of Mathematicians in 1995. So many awards. My connection to Professor Lee Lorch came through my own academic supervisor, Professor Edward Twizel at Brunel University in London, who was a former student of Lee Lorch's at York University. And this is Lee Lorch here receiving it's at AMS joint meetings in 2007, I believe, in New Orleans, receiving the Distinguished Academic Award. And also here, this was a major award he received in Canada by the Canadian Association of University Teachers 2012 for it's a Distinguished Academic Award for Excellence in Research, Teaching, and Services. And this award was now later renamed after Lee Lorch for his excellence in service research, advocacy, and so on. In fact, I remember just a few weeks after the award was delivered to him, he couldn't attend the actual ceremony in Ottawa to receive the award. So it was delivered to him in his residential home, and I visited him like a few weeks afterwards, and he was so proud of this award. He was pointing to me the minute I entered his room. He was pointing to that, and it's so fitting that it's renamed after him now. Okay. Yeah, and this is Professor Edward Twizel, my former advisor. I was showing him my first book that I edited in his office, and this was Lorch the day that I met with him when he got the award for the Canadian Association of Universities. And this was him visiting us a while back. Yeah, anyway. So the lecture today is Mathematics of Infectious Diseases. So the role of mathematics in providing insight into the spread and control of infectious diseases of public health importance. So I will start the lecture by stating that we as humans, have contributed a lot in the presence of many of the deadly diseases that we have been encountering. So our own activities, including habitat destruction, deforestation, agricultural development on wildland, climate change, environmental degradation, biodiversity loss, exposing disease-carrying animals to come closer to humans is primarily what's responsible for many of the disease outbreaks we've been experiencing since beginning of recorded history. So that's the outset. Right from the outset, I should say that. Okay, we as humans are doing certain things to the environment, to the landscape that is making us vulnerable to the spread of many diseases. So I've given some examples here, like smallpox killed over 300 million people in the 19th century alone. Black plague, over 75 million people. HIV/AIDS has killed over 35 million people. Currently, 38 million people live with the disease. And so on. TB, tuberculosis, is killing 1.5 million people around the world each year. Malaria used to kill 1 to 2 million people each year. Now it's killing about 400,000. This is largely due to the large-scale use of insecticide-based interventions, which we'll talk about later on. COVID-19 came in 2019 and already killed over 2.7 million people. Okay, so there are other major outbreaks that occurred also, like the multiple pandemics of influenza. We have the H2N2 pandemic, 1957-58, and the more recent one was the 2009 swine influenza pandemic, which started out of Mexico and I think the United States in 2009. And then the Ebola outbreaks in 3 countries in Western Africa, Liberia, Sierra Leone, and Guinea. Okay, so we've been having this, and you may ask the question, well, why do we care if diseases are happening in faraway places? And the reason simply is globalization. Okay, we're all vulnerable to what's happening in faraway places. I remember the Canada's Advisory Committee on SARS in 2003 made this quote that we're constantly a short flight away from serious epidemic. Actually, that's how SARS in 2003 came to Canada. A family went back to native Hong Kong and came back infected, and that's how it started. So we're all in this together, globalization and public health. The further we travel around and we do global trading and so on, make us all vulnerable to what's happening in faraway places. But mathematics has a long history of contributions in the theory or the study of infectious diseases, going back to the pioneering works of Daniel Bernoulli, a member of the famous Bernoulli family of theoretical physicists and mathematicians. And this Sarono Rose, Bernoulli's work was on assessment of immunization strategies against smallpox. And smallpox happened to be one of the only 2 vaccine-preventable diseases we have eradicated. We're close on the eradication of polio. We just have 1 or 2 countries trying to eliminate those places. And I'm sure the WHO will soon declare polio to be eradicated. This Sarono Rose was a British surgeon, a polymath, a poet, a mathematician, a novelist, who first elucidated the entire life cycle of malaria mosquitoes, parasite, Plasmodium. And then Cormac McKendrick, those were Scottish biochemists and military physicians who formulated essentially the mathematical theory of epidemics. Okay, so it's a collaboration. When we use mathematics to understand diseases, it's a collaboration between mathematicians and practitioners in other fields or researchers in other fields. For example, we work with immunologists or epidemiologists, ecologists, and so on. So public health people, public health policy people. So they're interested in, for example, during spread of outbreaks of new diseases like COVID-19, they wanted early estimation of epidemiological thresholds and disease burden, okay? And assessment of intervention strategy to determine which ones will be most effective, cost-benefit analysis, optimal allocation of limited resources, like we have right now with vaccination, shortages. If we don't have enough, okay, how do we optimally use the limited resources we have to get maximum impact? Computation of herd immunity threshold, and so on. But mathematicians and computational scientists and so on also have their own wish list. We design mathematical models to try to provide insight into what is going on. So we need to study those models to gain as much information as we can in terms of their qualitative properties. So asymptotic stability, replication theory, and so on. So we can use those properties to inform public policy based on those mathematical insights generated. And there are also numerical components because some of the models we deal with are typically large and so multidimensional and nonlinear, therefore we may not be able to find solutions in closed form. We have to use numerical approaches. And that's a large deal of statistical component to this because we have models with so many parameters, so many state variables, so we need to fit the models with data. We do parameter estimation, we do uncertainty quantification, sensitivity analysis, which I'll talk about later on. The point I'm making here is if you want to address the world's most important problems, okay, we cannot do so in isolation. The era of working in a silo is long gone now. We all have to work in collaboration with scientists from other natural and engineering and even social sciences together. Addressing the world's most important problems require the coming together of people from all facets of science and technology. So that's the main point I'm trying to make. Okay, so this classical model for the simplest model maybe one can come up with for the spread of infectious disease is one in which the total population, let's say N of T, is split into 3 mutually exclusive compartments based on the disease status. So S of T are those who are susceptible to the disease, which means healthy people who have not been infected with the disease yet. So that's S of T, and I of T are those who have the disease already and are ready to transmit the disease to other people. And R of T are those who have recovered from the disease. So for a simple model, for this classical model, SEIR model is called, if a susceptible interact with someone infected, there's some probability infection can take place, and that probability depends on how many contacts that susceptible person has with the infected person. So if this is an effective contact, so beta is an effective contact rate, which means contacts capable of leading to infection. So if the infection happens, we have a negative term here, the person is no longer susceptible, he or she is infected, so it moves to the infected class here at the same rate. And an infected individual can recover at a rate gamma and then move to the recovered class. So for this model, these are the graphs of this. We plotted this. So these are the rate of change. These are differential equations, the rate of change for each of these 3 compartments. And we plot each of those compartments here. So in formulating this model, we have to make fundamental assumptions. Just like in every other model, if you want to formulate a model, you have to make assumptions. Okay, so we assume the population is closed, nobody comes in, nobody leaves, and that the population is homogeneously mixed, which means every member of the community is equally likely to mix with every other member of the community and acquire infection in that way. And the waiting time in each compartment is exponentially distributed. And once you recover, you don't get the infection again, and so on. So there are some, another thing, yeah, this is an epidemic model. Therefore, we assume that the time scale of the disease is much lower or smaller than the demographic time scale, which means we assume that the disease is going to be around, a new disease maybe for a year or 2, but the demographic time scale in the US, for example, people live on average to be 78 years, 79 years. So that's why it's called an epidemic model. We do not have birth rate and natural death rate in the model. Okay, so, and this makes sense for a new disease. Okay, but of course there are many extensions to this basic model to include so many different heterogeneities like age structure, like risk structure. Some are more at risk of acquiring infection, spatial structure, and maybe mixing patterns. People mix preferentially, maybe with their friends, with their colleagues at work, and so on, people within their social networks and so on. And you can also include some interventions on therapies and multiple strains, or anthropogenic climate change, environmental changes, land use changes, zoonosis, and so on. Okay, so this is the basic framework that I'm going to be working on. So you can extend this model, for example, to do an SEIR model. So in the previous model, we showed that once you get infected, you are ready for prime time. You are instantaneously capable of transmitting the infection to other people. But in reality, of course, in many disease situations, once you get infection, there's a time lag before you become infectious. So that's the period we call E, the latent period or the exposed period. And here we include demographics. So it's for a disease, this model is for a disease that has been established. So we have birth rate, like rate at which new recruitment rate into the population, either through birth or through immigration. And we assume it's fairly constant, pi. And susceptible individual interacts in infectious fraction, I divided by N. There's this probability of infection and this effective contact rate beta. So if infection happens, you go to the E class. And mu is the natural death rate. So 1 over mu is the average lifespan in the community. So, so sigma, if you're in the E class after a certain period of time, 1 over sigma, the incubation period typically, you become infectious and move to the I class. And then you recover at rate gamma and die naturally. Okay, so this is an SEIR model. And so it has 4 compartments. And we need to study this in a region where the model makes biological and physical sense. Because we're dealing with human populations, all state variables of the model have to be non-negative, right? So we have to study the model mathematically in this cone here, where as the total population in the absence of disease actually is pi by mu. So the sum of all 4 compartments cannot exceed pi by mu. So that's essentially the carrying capacity. And we can compute the reproduction number. The base. So you've heard so much about reproduction number during COVID-19, R0, right? Everybody has heard of R0. So it is essentially the average number of new cases generated by a typical infected individual if introduced in a completely susceptible population. So if I'm infected, I'm introduced into my classroom, for example, and nobody in my classroom has the disease. How many of my students am I going to infect before I get cured of the disease, or if it's fatal, before I die of the disease? Hopefully I don't die of the disease. Anyway, so that's what R0 is. So typically, by first principle, R0 is the infection rate near the disease-free equilibrium times the average duration of infectiousness, which means my ability to infect somebody, okay, as a susceptible person, times how long I'm infectious, how long I'm able to infect the person. So here, the infection rate, my ability to transmit the infection if I'm in I interacting with S is beta times S star divided by N star, where S star and N star are the components of S and N at the disease-free equilibrium. And I have to be able to survive the E class. My probability of surviving the E class and going to the I class is sigma divided by the sum of the linear terms leaving the E class, which is this term. And because we assume exponentially distributed waiting time in compartments, we have the average duration in the I class is 1 over gamma plus mu. I had a delta here, that's a typo, I should not have the delta. Anyway, it's 1 over gamma plus mu. So that's the R0. The reason this is important is that's what public health people want. Okay, if they want to control the disease, what they need is to do whatever it takes to bring R0 to a value less than 1. Because if on average, I cannot transmit the disease to more than one other person, then typically the disease will die out with time. So you can see that from here, S star divided by N star for this model is 1, because in the absence of disease, the subtotal population is pi by mu, the total population is also pi by mu in the absence of disease. So we have just beta times sigma over sigma plus mu times this term. So if we reduce beta, we're going to reduce R0. And you can reduce beta by doing things like contact tracing, like social distancing, face mask usage, and so on. And so by computing this R0, we're able to suggest to public health people what sort of strategies will make a lot of sense, because a good strategy is one that reduces R0 significantly. So that's the first thing. Okay, and so mathematically what we've been able to show is for this SEIR model, the disease-free equilibrium, which means equilibrium in the absence of the infected classes E and I, is globally asymptotically stable, which is a jargon for saying we're going to actually reach that equilibrium, okay, whenever the reproduction number is less than 1. But if the reproduction number is greater than 1, we go to a positive equilibrium, okay, when R0 is greater than 1, it's a stable endemic equilibrium. So the disease will basically persist in the population if the reproduction number is greater than 1, but it will be eliminated if it's less than 1. Okay, so what that means, if you implement a control strategy such that you are able to bring and maintain R0 to a value less than 1, then you will eliminate the disease from your population. In other words, R0 less than 1 is necessary and sufficient for the elimination of the disease. That's what it means. So this is a bifurcation diagram. It tells us that the dynamics of the disease is characterized by this number R0, right? The average number of new cases generated by a single case introduced in a completely susceptible population. So if R0 is less than 1, we have this blue region here, which is stable DFE, disease-free equilibrium. So we basically will controlling the disease here if R0 is less than 1. But the minute R0 becomes greater than 1, we have persistence of the disease, and we have a stable endemic equilibrium. And to the right of R0 equals 1, the disease-free equilibrium loses its stability, but a new equilibrium becomes stable. So that's exchange of stability from one equilibrium to another, and this is called a forward transcritical bifurcation mathematically. But the point is, if we are able, through intervention, to bring R0 to a value less than 1, we have a chance to eliminate the disease. For this model, SEIR, we are guaranteed that the disease will die out with time if R0 is less than 1. But if R0 is greater than 1, then the disease becomes endemic in the population. And we have certain diseases that are endemic, like malaria, like HIV/AIDS, and so on. But there are diseases we have eradicated, like smallpox, like rinderpest, for example, as I mentioned, using a vaccine before. Okay, and this is R0. This is a picture very nicely sent to me by Kamal Bali. Kamal, thank you for this. To illustrate R0. If R0 is 2, so the index case, which means the very first case is red, right? This is the person who brought the disease to the population. Everybody is black, so black means susceptible. You're healthy and you are vulnerable. You can get infected with the disease if you come in contact with somebody who has the disease, depending with some probability. So if R0 is 2, it means one red will generate 2 other reds, and so on. So you see, and this is the not, the 0 in R0 is to show that we're dealing with a completely susceptible population, which means nobody is vaccinated and no public health intervention is implemented, no social distancing, no masking, nothing. So if R0 is 2, you see that, and nothing has been done, all blacks will become red. But here we have a nice mixture of black, so which are the healthy ones, red, the infected ones, and the blue are the ones that have some kind of partial immunity, because either they have seen the disease before they recovered from it, therefore they have partial protective immunity, or that they have been vaccinated against the disease, and so on. So it's a nice mixture of what's going on. So this is called the control reproduction number, because here, unlike in this case, we have control being implemented by public health people. And the way to calculate reproduction number, there's excellent theory by Otto Dickmann and Histebik and Mertz, great paper written in 1990, to actually show you the next generation operator method to compute the R0, mathematically speaking, is essentially a spectral radius dominant eigenvalue of some next generation matrix. So that's how it's computed. And also my colleagues, Van den Drisch and James Watmore, formulated another way to, or essentially a similar way to compute the reproduction number. Okay, and there are many different types of models. The one I just described is compartmental, is deterministic, but you can also make it stochastic, you can make it discrete time, spatial, and so on, or you can use agent-based or contact net. So the type of model you use depends on the question you want to address and also the availability of data. For example, if the question is to determine who infected whom and when, then it makes a lot of sense to use an agent-based model because it keeps track of individuals as agents, right? How individuals interact with each other and how they interact with the environment and so on. So the type of model we use depends on the question we want to address and the data that we have. Agent-based model is data hungry. You have to have a lot of data, and it's computationally intensive as well. You have to, but anyway, those are different types of models. There may be others also that I did not include. Okay, so I would give 2 case studies for this lecture. One is vector-borne disease, and the other, of course, naturally, COVID-19 that we were talking about. So vector-borne diseases, these are diseases caused by vectors, okay, like mosquitoes, for example. And they account for 17% of all infectious diseases, okay? They cause about 700,000 deaths each year. Before that, malaria alone was causing 1 to 2, before 2010, 1 to 2 million people deaths each year. So malaria affects 1/2 of the human race. You know, it's generated about 219 million cases each year, 400,000 deaths annually since 2010. Dengue is even more spread, highly prevalent. 96 million symptomatic cases, about 40,000 people are dying of dengue disease each year. We have the dengue mosquitoes here in the United States, Aedes aegypti, Aedes albopictus, and we're surrounded by countries in Latin America that have sporadic outbreaks of dengue. So because of mobility of people, of course, we're vulnerable to getting diseases like dengue and Zika and chikungunya. We saw that already in Miami. Because the mosquitoes are already here. All you need to kickstart an epidemic is a few people go to the endemic areas and become infected and come and infect their local mosquitoes. Anyway, there are other vector-borne diseases that are listed here. But I'm going to focus on malaria because there's a big push now to eradicate malaria because of the huge success made using insecticide-based intervention. So it's caused by this parasite Plasmodium, and there are many species of the 5 of them, at least 5 of them. And one of them, falciparum, is responsible for over 90% of the death, okay, mostly in sub-Saharan Africa. So, and there are about 450 different species of the vector, the Anopheles mosquito, of which 41 are major vectors. To be a major vector, Anopheles species has to satisfy 3 properties. One is it has to habitually bite humans, okay, because the transmission is to humans. It has to be able to carry Plasmodium, okay, and it has to live long enough to complete the sporogonic cycle, which makes which means when the mosquito gets infected, how long it takes for it to become infectious, okay? So those are the properties, and there are many species. Gambier is the dominant one, and it thrives in situations whereby anthropogenic alteration of the environment, such as deforestation, construction, livestock, occurring. And so that's why we have a large outbreak or large burden of malaria in sub-Saharan Africa because of the predominance of Gambier and so on. Anyway, so the potential vulnerability in the US, as I alluded to earlier on, Anopheles is already here, maybe not necessarily in large numbers, large abundance, but because of climate change, because of host mobility to and from endemic areas, just like we've seen with Zika, we're vulnerable to mosquito-borne diseases, including malaria as well. Okay, and yeah, so forest, clearing forest makes us vulnerable, creating breeding habitats for mosquitoes. That is a nice quote, that it's only when man cuts down the forest that breeding places for Gambier become almost infinite, and just classic illustration of that situation. Okay, so typical life cycle of malaria mosquitoes is when a new mosquito pops out, okay, it looks for a human host to bite. When it's succeeded in biting, it goes, so that's the stage one of the gonotropic cycle. The gonotropic cycle is a host-seeking cycle. Okay, so it looks for a host to bite, succeeded, goes to stage two to try to rest for the development and maturation of the egg, which is temperature dependent. And when that is done, then it goes to stage three, looking for breeding habitat, which is a body of water here. So Anopheles mosquitoes require water to lay eggs. And once the eggs are laid, they go to those instar stages, the larval stages, stages 1, 2, 3, and 4, based on the size of the larvae. And then the larvae become pupae and then become an next adult mosquito, and the cycle continues. So this is typically the life cycle of the mosquito. Okay, so a model for mosquito-borne disease like malaria should, at the very least, capture some of the stages. Okay. All right, so Plasmodium has been around for millions of years, not in humans, but in non-mammalian primates like lizards for about 130 million years. Falciparum crossed over to us reportedly from gorillas some 12,000 years ago, toward the end of the last ice age and the beginning of the era for global warming and the subsequent human agricultural civilization and land use changes and so on, concentration of human settlement. This is what was responsible for human malaria, okay, because of all the things that we as humans were doing to the environment, okay? And climate affects all aspects of the mosquito life cycle, pretty much the aquatic stages, the adult stages as well. So for example, due to increases in temperature, bite rate increases, faster blood meal digestion, faster immature development, immature mosquito development, and so on, and survival as well. Okay, so the simplest malaria model I can give here, and I'll quickly run through it, is the Ross-McDonald model, the classical Ross-McDonald model. So the total population of humans is H of T, the total population of mosquitoes is M of T. It just considers two populations, infected mosquitoes, X here, and infected humans, Z here. And this is the rate at which infected humans acquire infection. And this is the recovery rate of humans R. And this is the natural death rate of mosquitoes. And using this simple model where A is, what is A? A is the mosquito bite rate, B is the probability of infection from mosquito to human and from human to mosquito and so on, R is the recovery rate. And this is the reproduction number. The reproduction number for this system is given by this MA squared BC divided by mu R. And it's clear from here, what you need for control of malaria for this using this model is to reduce M, okay, or to reduce R naught. See, M is in the numerator, reduce M. M is the host vector ratio, how many mosquitoes per human. So you reduce that, you reduce R naught. And that's what Saruno Rose used to suggest to the WHO to focus on larval habitat reduction. Okay, larviciding. That was the main control strategy against malaria in Western Europe during that time. But later on, George MacDonald came along and said, well, okay, well, this model does not include the fact that once the mosquito gets infected, it does not become instantaneously infectious. It needs to survive the supragonic cycle, okay, to become infectious. So he added that time delay in the model, and that led to the formulation of a new strategy based on focusing on adult mosquitoes, adult survival. So instead of trying to kill the larvae, the eggs, and so on, just wait until the mosquitoes have become adult mosquitoes, and then you kill them using indoor residual spraying and so on. So DDT typically was used at the time. Yes, so it was invented, DDT was invented in 1939, was used during the Second World War, and it was the basis for the aim to eradicate malaria. But it was disbanded in 1969 because the mosquitoes have become resistant to DDT. Okay, so a lot of success between 2000 and 2015, largely due to large-scale use of insecticide-based interventions, indoor residual spraying, and long-lasting insecticidal nets. So these are bed nets that are used in endemic areas, so people sleep under them. The idea is the bed nets have two efficacies. One is the killing efficacy. They are impregnated in highly toxic material, chemical, so that when the mosquito lands on the bed net, the mosquito instantly dies, depending on the efficacy of the bed net. And the bed net also has the deterrence efficacy to deter mosquitoes away because the human is shielded. But majority of the reductions in malaria cases and burden, about 81% was due to the long-lasting insecticidal nets. The Gates Foundation donated 1.5 billion bed nets to malaria endemic areas between 2000 and 2015, I believe. Yeah. So that generated a lot of success, but unfortunately there's now stumbling blocks to the new quest for eradication, one of which, of course, is the resistance to all currently available insecticides that are used in IRS and long-lasting insecticidal nets. So the mosquito is resistant, meaning if it lands on the net is not going to kill it. Okay, it may deter it, but it's not going to kill it. Okay, depending on the efficacy again. So this is a mutation that happens inside the mosquito that makes them to resist the toxin that is embedded in long-lasting insecticidal nets. Okay, so now climate change, of course, as we have already seen clear manifestation of it, the Eastern Highland malaria. This is in Eastern Africa. They didn't used to have malaria before, but now because of global warming, they're much cooler region before, but now it's getting warm, it's warmed up, and they've been seeing an increase in malaria since the 1980s. Parasite resistance to drug therapy, of course, artemisinin is used to treat people with malaria, but there's evidence now that in certain places like in Cambodia, Laos, in Vietnam, that there are cases where the plasmodium have become resistant to the drug therapy. And of course, mobility of humans, so rural-urban migration, land use changes, and so on. So the question is, and there's no safe and effective vaccine against malaria, although there are some that are using an mRNA vaccine that's undergoing clinical trial in Tanzania at the moment. So hopefully that will lead to some big success. So the point is there's a huge need for mathematical modeling and analysis coupled with statistical data analytics to provide realistic insight into the effects of all of these stumbling blocks for the effective control of malaria, and to try to see whether or not we can still achieve eradication based on all of these things. Can we find a way using existing infrastructure, like based on existing insecticide-based interventions to eradicate malaria? That's a big push now to eradicate malaria, but these are the things that get in the way. Can we still achieve that? And I think that's a major role of mathematical model. And I'm going to just describe a few of these options. So this is the current malaria map. So red is where malaria is happening. Yellow is where malaria transmission is happening in some places. Red is everywhere. Blue is unknown, and so on. Okay, so one key question to ask is, will this map change if there's 1 degree increase in global temperature due to global warming? Are we going to start seeing places like Australia here that has basically no malaria to start seeing malaria because it's surrounded by patches of malaria here? Okay, or even here in North Africa, I mean, Central Africa, malaria, but there's mobility of people. So these are the kind of questions we can try to address. Yeah, so I've listed here some key modeling questions that can be addressed. Here, can we, and this is probably one of the most important questions right now within the malaria ecology community. What is the link, if any, between insecticide resistance and malaria epidemiology? Does insecticide resistance, again, insecticide resistance means the mosquito is now resisting the insecticides that are used in indoor residual spraying or in bed nets. Okay, is there a connection between resistance and malaria prevalence? Does resistance make prevalence go high or lower? So these are questions, these are open questions that have not been answered, and we're trying to address those questions. So you're trying to quantify the impact of fitness cost of resistance, because if the mosquito is resistant, there are a lot of fitness costs in terms of, for example, survival of the mosquito, ability to mate, ability to reproduce, and so on. So we have to work here with entomologists to carry out the lab and field experiments to find good estimates for those fitness cost related parameters. And this is work I've been working with some of my colleagues here at the School of Life Sciences at Arizona State University. So the key question is, can we eradicate malaria using existing insecticide-based intervention? Can we quantify the impact of climate change on the mosquito and the parasite? Can we also quantify the impact of host and vector mobility? And also resistance, parasite resistance to combination therapy and so on. And co-infection between malaria and other pathogens, including now COVID-19. How is COVID-19 going to affect malaria incidence or prevalence in endemic areas? And this is a really key question, big question. Okay, so we started with a working group on climate change and vector-borne diseases funded by NIMBIOS, National Institute for Mathematical and Biological Synthesis. I forgot to acknowledge at the beginning the sponsoring agencies, but NIMBIOS sponsored our working group on climate change and vector-borne diseases. And NIMBIOS was a great place. It's a great place to do mathematical biology. They provided really excellent quality training and research programs and collaborations. Since its inception, now they're making some transition. Lou Gross has been an excellent director. And now the transition is Nina Pepperman here is going to take over a scale version of NIMBIOS. And I'm sure they continue to do great things. Anyways, this is a working group. We started working on the impact of climate change and vector-borne diseases. And now I give a simple model for analyzing the impact of long-lasting insecticidal illness and climate change. So here, the setup is, the model is complicated. The setup is the following. We have a mosquito that comes in, and we have the 3 stages of the gonotropic cycle. So each stage, the mosquito in each stage of the gonotropic cycle, stage 1 is SEI, either the mosquito is susceptible, either is exposed, or is infectious, SEI. And X is the first stage, Y is the second stage of the gonotropic cycle, and Z is the third stage. So once the first stage, the mosquito is looking for a human to bite, the human may be sleeping under a bed net, in which case there's high probability that the bed net will kill the mosquito and then it dies, it doesn't proceed, or it succeeds in getting the blood meal from the human. The mosquito needs the blood meal to lay eggs, basically, for the process of development of eggs. It's the protein in our blood that the mosquito uses to process egg development. So if it succeeds, then it goes to stage 2, which is SY. It's still susceptible because this human is in the net, it's not infected. And once that happens, it goes to stage 2, rested and goes up towards stage 3 and lay eggs, and the eggs become larvae, all the 4 stages to pupae and so on. Okay, so, and you can go the other way around. This mosquito bites an unprotected human, which means a human not sleeping under a bed net. It's possible the human can kill the mosquito by just snapping their hands on the mosquito, or that the mosquito succeeds in taking the blood meal and go to stage 2 and then continue to stage 3. And it's also possible the human that the mosquito encounters is infected, which means it's E class, and then he goes through the same stages as well. So this is a model, and I was going to say the Ross-McDonald model has only 2 equations, but as a very wise man says, everything should be made simple, but not simpler. That was Albert Einstein. So because if you want to really address the impact of long-lasting insecticidal nets and climate change, you need a model structure that has all of this. Because the gonotropic cycles, the immature dynamics are very much affected by climate change, particularly changes in water and air temperature. Okay, so we have to expand the model, Ross-McDonald model, to include some of these compartments. But then it comes with all its complications, because the model has about 22 equations, many parameters. So obviously it's difficult to do analysis, it's difficult to do statistical and computational. So it's difficult to do theoretical analysis. Of course, we can do bifurcation analysis, we can do existence stability, but we are not able to find exact solutions in closed form. So there are challenges also with statistical parameter estimation, identifiability issues, uncertainties in the estimates of the parameters, computational. So there are papers here I've listed by Salih Blow and Dola Travati on sensitivity analysis, classic paper, and a great book here written by my colleague, my friend, Maya Marshall at University of Florida on mathematical epidemiology, which addresses many of these issues, particularly identifiability. And my colleagues here, Professors Kuang, Nagy, and Eikenberry also wrote an excellent book on mathematical oncology, which addresses many of the statistical issues in regards to cancer dynamics. So I recommend this book. But the point is 22 equations, we cannot find solutions in closed form. So computation is a major component of this. So how do we design numerical methods that capture the qualitative dynamics of the continuous time models that we use to study this transmission dynamics of diseases? So that leads us naturally to the work of Ron Mickens at Clark Atlanta University, distinguished fellow professor of physics, and who coined this non-standard finite difference philosophy. The idea is you want to design numerical methods that are dynamically consistent, which means they are capturing the correct dynamic properties of the continuous time models. Okay, and those are the kind of methods that we should be using rather than black box methods inbuilt in ODE 45, for example. So this is his latest book that came out last year on applications of non-standard finite difference method. And this was a book we did, AMS special session to celebrate his 70th birthday. So I edited this volume. This is Ron Mickens in the middle here, receiving his Blackwell Tapia Award 2018. And this is Professor John Labuma from the University of Pretoria in South Africa. This is Talitha Washington, Professor Talitha Washington, also at University of Clark Atlanta University, and Professor Ron Bachmeier from Occidental College. And John Labuma and his colleagues at University of Florida, particularly Professor Ruman Angelo, they formulated this mathematical analysis of non-standard finite difference scheme. Actually, they mathematized the whole process through their coinage of elementary stability properties of non-standard finite difference methods. So I recommend some of these papers. And what we show from this model, which is very important, is that it has a backward bifurcation property. So which means, what it means is, instead, unlike in the previous case where we have the forward bifurcation, when R0 is less than 1, we go to the disease-free equilibrium all the time, and it's greater than 1. No, here we have coexistence of two stable attractors. The bifurcation at the bifurcation point is backward. We have the coexistence of two stable attractors, okay, where, so it depends on which equilibrium you go to, depends on the initial conditions, okay? So if the initial condition is above the stable manifold, the saddle point, we go to the endemic. The point is, even if reproduction number is less than 1, we still have the disease persistent. So this is one property that was shown to happen with mosquito-borne disease. And I think Dr. Salis Garba of the University of Pretoria showed for the first time, I believe, that disease-induced mortality causes backward bifurcation in vector-borne diseases. This is a paper I recommend to use. Okay, so now having done all this, we can go now to try to look at the impact of climate change on population abundance of mosquitoes along lasting stratum, particularly in Africa. So here we show the correlation between parasite rates in many parts of Africa with the R0, the reproduction number, and we show that it's actually correlated very nicely. Places with high parasite rate, they tend to have higher R0, okay? And here we also see that the malaria map, okay, so if there's 1.5 degree increase in malaria in global temperature due to warming, what is the impact in Africa? And we see here, so red means you have malaria, and you see expansion of malaria and going eastwards. And this is, we cycle this area here. This is Eastern Africa, consisting of large populations like Uganda, Tanzania, Kenya, and so on. So we see movement towards, because that's the question I was asking earlier on, what is the potential impact of global warming on the distribution of malaria mosquitoes and malaria disease in endemic areas? And clearly this shows with global warming, we're going to have more cases and expanding eastward. So, and this is again what's happening with in Eastern Africa, the whole region of Eastern Africa was 1.5 degree increase. You see some of this that are yellow, low malaria, but now they're having more malaria. And so on. This is for the 2018 projections, regional climate projections 8.5, and again, much more. So the point is, for the interest of time, I'm going to skip something, but we're having more and more malaria because of global warming, and also the efficacy of the bed net. So here, this is a baseline case when there's no bed nets used, but we have a super duper bed net with slow resistance, which means bed net is really highly efficacious in killing the mosquito when it lands on it. And you see that most of the yellow become basically blue, no malaria. So clearly the efficacy of bed net to do this. So, and of course, if things get worse if you increase high resistance, which means the bed net is really not killing the mosquitoes, and also increase regional temperature due to global warming. So you start to see more malaria. Okay, and we also calculated what is the optimal temperature for maximum malaria activity, and it's clearly about 30 degrees in most of these places in Africa. The point of this is if you are in a place where the mean monthly temperature is less than 30 degrees, but global warming moves you up to 30 degrees, you're going to be seeing an increase in malaria burden. And if you are above 30 and then you have more warming, then your malaria burden goes down. Okay, and this is what we're already seeing in Central and Western Africa already. We've seen a decrease in malaria burden, but we're now beginning to see increase in Eastern and Southern Africa. Okay, so for this interest of time, I was going to do genetics epidemiology modeling framework, but I really don't have time. I have to run to the COVID stuff. What we showed here is that there's a control window. For this work, we incorporated or included the genomics of mosquitoes, okay? So, so we had the mosquitoes split into those that are resistant to insecticide and those that are not. And we found condition in parameter space where we can have effective control window using both IRS and long-lasting insecticidal nets. So we found maybe bed nets. If we're using only bed nets alone, if 90% of the population can use bed nets, the really good quality ones, we can eliminate the mosquito, the malaria, but otherwise it's going to be very difficult and so on. So we found this in this window, we can eliminate, otherwise it's difficult. And we had also biological controls, such as the use of CRISPR-Cas system, SIT, to modify the genetic makeup of the mosquito to produce the mosquitoes we want. For example, we want to modify the DNA of the mosquito to make it not amenable or not able to have a malaria parasite. Or through the sterile insect technology, we release genetically modified sterile male mosquitoes so that when they mate with the adult female mosquitoes, the eggs laid by the adult female mosquitoes will not hatch into larvae. Therefore, you reduce the mosquito population abundance by doing that. So there are a lot of a number of biological controls that are implemented or that are suggested for use against malaria. The motivation is simply because now we have this insecticide resistance to all 5 chemicals that are used in IRS and bed nets. So which means that's potentially a problem for eradication, of course. And we don't have a vaccine. So we need to look for other controls, and it takes years to come up with new chemical insecticides. Okay, so then we have to look at biological controls. But the biological controls also have their own issues because logistically, you have to rear these mosquitoes in the lab and deploy them and so on in the right place. So anyway, so we have a simple illustration of one case. This is a sterile insect technology. There's a drone here. We're releasing those mosquitoes that are genetically modified, male mosquitoes. Okay, so these are and then they mate with, so the blue ones are the sterile males that we have modified and released, and the brown ones are the wild male mosquitoes, the ones that are not modified, and the green ones are the unmated females, adult female mosquitoes that have not mated yet, and the purple are the female mosquitoes that have mated with the sterile male mosquito, but they will be laying eggs that will not hatch. And then these are the possible outcomes. Okay, so we model this to see whether or not this strategy will work. Can we reduce the population abundance of mosquitoes using this strategy? And this is the model we use, and it actually shows that, so the mosquito abundance is reached actually at 30 degrees Celsius. Here we can see that the various mosquito populations that we do not want die out immediately. We did the simulations for the first 2 years, and then we release the sterile male mosquitoes, and then the population of the wild mosquitoes immediately goes down. But then the sterile mosquitoes survive. And that's what we wanted. Okay, so I now have to run to COVID-19. It started in 2019, spread to pretty much all countries on Earth. By March 11th, the WHO declared it to be a global pandemic. And it came, so again, as I mentioned before, our proximity or our interaction with non-human primates led to all sorts of diseases, including the swine influenza pandemic that I talked about. And the birds are normally implicated for this. This is the SARS of 2003, which started out of civet cats, found civet cats, and to humans. and so on. And so COVID-19 came out of pangolins and so on. So I compared the three different coronaviruses. SARS started out of Guangdong province in China, MERS started out of Saudi Arabia and Jordan, the Wuhan, COVID-19 out of Wuhan. And look at the mortality. The case fatality ratio is 9.5% for SARS, 34% for MERS, 2.2% for COVID and so on. Killed millions and millions of people. And this is the burden in the US for the three waves that we have seen. This is the daily confirmed cases. This is daily death in the United States. And I compared 10 different top 10 countries in terms of mortality. Notice here in Taiwan, there are only 10 deaths, only 990 confirmed cases. And we have 3 vaccines, actually 4 vaccines now that have been approved for use, 3 by the FDA, 1 by UK, in the UK. The first 2, Moderna and Pfizer, were mRNA based, and the Oxford one is inactivated virus based, is AstraZeneca Oxford. They have different efficacies, different cost, and different modes of implementation, 2 doses each one for the 3. And now recently we have the Johnson & Johnson vaccine that have been implemented. So 2 million vaccinations per day right now in the United States. How will this impact the dynamics of the variants that are going on? Okay, so there's lots of questions one can ask using modeling in this context of COVID-19. For example, let me see if I can. Yeah, so we can assess the impact of early implementation of control strategies. Can we focus the typical cause of the pandemic, zoonosis, epidemic intelligence, impact of pandemic variants on the burden and vaccination programs, competitive exclusion principle? Will the new variants dominate and remove or exclude the dominant strain through competitive exclusion? Can the variant drive it out to extinction? Okay, so those are some of the questions and that we can address using modeling. And this I've done with my collaborators here, my former student, Anaharo, postdoc, research associate, Professor Eikenberry, and Callistus Ingongala from University of Florida, to my 2 students, Marina and Samantha, and then this is Matthew Scotch, molecular epidemiologist at our Biodesign Institute. And we also did the MSRI at joint workshop, but for the interest of time, I'm not going to go into that. But we did a workshop at MSRI. You can go to the website to get all the papers and presentations. Okay, so we fitted the model using cumulative mortality data for the United States for the 3 waves, and we got a reasonably good fit. Therefore, that served as a proof of concept for us to use the model to make predictions. Okay, so the first question we ask is, what happens in the United States if we started masking right from the very first day or first few weeks of the outbreak of the disease? And we use 3 different types of masks. So there's this one, the cloth mask, which is just single layer, not very effective, or 30% effective. Improved cloth mask, which is about 50% effective. The surgical mask like this, which is about 70% effective. And then the really good mask, N95 mask, which is 90% effective. So we're able to show that clearly if we started early. Okay, see what's happening now. So the green is the N95 respirator. This cyan is surgical mask, magenta is improved cloth mask, and green is cloth mask, and so on, and baseline is blue. And you see that if, for example, we started here, this is B, we started on February 18th, just 1 month after the index case in the United States. If we started using masks, okay, and let's say we use N95, if everybody is using N95 masks in the United States, the epidemic would not have even taken up in the US, essentially, we are here. Or if you use the other one, the surgical mask, we would have an epidemic, but it's not going to be as devastating as it was. So its early implementation of control strategies will make a huge impact. And this clearly has shown that masking, if we started masking at the beginning, would have made a huge impact. So here, we can look at, for different mask efficacy, if we take the 30% efficacious mask, can we bring R0, the reproduction number, to a value less than 1? And the answer is no. But if we use N95 respirator, we need about 40% coverage. If 40% of the US population are wearing N95 masks at the beginning, we'll be able to control the disease immediately. Okay, and this increases with increasing levels of social distance. For example, if we increase, we're doing masking, but we also increase social distancing or other controls, non-pharmaceutical intervention, we do much better as well. Yeah, and this is early implementation of lockdown as well. If we started locking down early, we see this levels, and we have actually predicted, this was the work we did, which came out in August, and we predicted the third wave of the pandemic based on what was happening in the states of Arizona, Florida, New York, United States. Third wave around end of October, beginning of November. Okay, so we did, for the interest of time, I'll skip the vaccination part. I'll just show the, yes. So we showed here, using the 3 different types of vaccines, can we achieve herd immunity in the United States? Herd immunity is achieved using the AstraZeneca vaccine if 80% of us are fully vaccinated with AstraZeneca vaccine. But for the Pfizer and Moderna, we showed that it's about 59%. About 60% of Americans, if they are fully vaccinated, we achieve herd immunity. And the level required, the herd immunity threshold decreases was again increasing baseline levels of other interventions. If we combine vaccination with increase in social distancing, the level of herd immunity threshold decreases from 60% to about even 40% if we can increase social distancing baseline at the beginning of third wave by 30%. If more of us will continue to mask up and socially distance, even though we're vaccinating 2 million vaccines a day, we can achieve herd immunity by, which is more realistic, we can 40%, we just need to vaccinate 40%. But fully vaccinated meaning you get the 2 doses if it's Pfizer and Moderna, or if the 1 dose if it's Johnson & Johnson. Okay, so yeah, so that's the main conclusion of this is, okay, yeah. So here we can achieve elimination of COVID-19 using the various type of masks, particularly N95, if we can achieve compliance, okay, of moderate compliance, 30 to 40%. That is definitely possible. So going forward, if we have a new pandemic of a coronavirus like this, if we've been able to stockpile this, N95 respirators, okay, and make sure we give everybody right from the beginning, maybe do the Defense Procurement Act, to massively produce this and make them available, you know, we would have saved a lot of lives and of course a lot of economic costs as well, because the burden of COVID-19 is just incalculable. You cannot just quantify how much was lost in terms of wages and in the economy. But if we started early using the right type of masks, we can eliminate easily, we can control it much easier. And we can get herd immunity if we are fully vaccinated at a rate 60% using Pfizer and Moderna, which are the 2 vaccines being used in the United States right now. And now the the Johnson & Johnson vaccine. We can actually eliminate, based on our estimation, sometime in the fall, if we continue on this trend of 2 million vaccinations a day using the Pfizer and Moderna vaccine, it's definitely possible to have elimination or near elimination this year. So this is quite promising based on our estimates. The point is, as long as we're interacting with non-human primates, we're doing all of the things, yes, we can use COVID, or we can use masking and social distancing and lockdowns and so on to control diseases, but it's just a band-aid solution. History is going to continue to repeat itself until we start taking the hard look at the problems that we are imposing onto the planet in terms of environmental degradation, biodiversity loss, and wildlife issues. We're going to be hit with this coronavirus, other pandemics as well, not just coronavirus. Okay, so we really have to look at the big picture, you know, and really do all the things. Okay, so history will continue to repeat itself unless we get our act together on this. Okay, so just to wrap up, I also like to mention some of the capacity building and training initiatives we have been doing in Africa. For example, the US-Africa Partnership, which was administered by DIMAX and funded by NSF in collaboration with MBI, Mathematical Biosciences Institute in Columbus, Ohio, AMS, Africa Institute of Mathematical Sciences in South Africa, SEIR, Southern Africa Center of Epidemiological Modeling. So we ran it with Fred Roberts, Professor Fred Roberts, director of DIMAX, very successfully. We trained lots and lots of people in Africa. Many have got their PhDs and work in various universities in Africa, and including also public health agencies, so they have the capacity they need to be able to formulate policies to control the spread of diseases in their localities. So the project is finished, and now my colleague, Professor Overton Janda at Auburn University, is running his Masamu project, also funded by NSF, and it's really good. We take students from the US, from Canada, to go and work with students in Africa in collaborations to do research projects together. It was really, really excellent. One of the most successful projects we're very proud of. And the AIMS project is Africa Institute of Mathematical Sciences, started by Neil Turok, And Neil is one of the most visionary people I know. He started the network of AIMS. The first one started in Muizenberg, South Africa, near Cape Town in 2003. And now we replicated that in 5 other locations in across Africa. In fact, he made this bold claim. He was a professor of theoretical physics at Cambridge, worked with Stephen Hawking, and then became director of Perimeter Institute of Theoretical Physics in Waterloo, Canada. And he announced this next Einstein forum because of the estimate by 2050, one in 4 people on planet Earth will be African. So Neil Turok said, well, okay, the next Einstein is going to be an African. Not only an African, but it's also going to be a female from Africa. That's very ambitious, and he's pushing on that. So he started the next Einstein Forum to try to help reach that goal. And he has been, so this is the former Prime Minister of Canada, Stephen Harper, announcing $20 million support to the AIMS project to replicate AIMS in other places out there. This is me somewhere here at the back. And Stephen Hawking was there. And this was a photo of at the Perimeter Institute in Waterloo. And this was us at Neil's apartment having breakfast before the actual formal ceremony. And this was Stephen Hawking, was AIMS student in Muizenberg in South Africa, just fantastic advocate for science, for AIMS, for STEM in Africa. And okay, so now to change the subject completely, and for the Monty Python enthusiasts, this is this heading from the BBC says, and now for something completely different. I should send you a postcard from Pretoria, University of Pretoria, where I have a joint appointment. This was us after one of the summer schools we run in Pretoria. This is a sleeping lion, okay, and out at the back there, and this is at the end of this summer school. And this is Aziz Yakubu from Howard University. This is at the back here. You see Alan Lloyd, North Carolina State. At the back right here is Fallen van den Drisch. They are all holding their baby lions. And that's really good to see. There are lots of students here and postdocs and researchers from around the world coming to work together. And this was me. I never thought I could do this, actually hold my own baby lion. But I did that and I survived. And even cheetah, okay? Anyway, so I will stop right here and thank you so very much for your time, and I'm very happy to take questions.
Okay, thank you, Professor Gumel, for a very interesting talk on a topic that, as your talk has illustrated, will unfortunately become only more and more important in the near future. One wishes it were otherwise, but I'm afraid this is the world we live in where we really need this work and more than ever. So we have had this has been a long and very informative talk there. So I would just like to ask relay a couple of questions from the audience. Several people asked, you mentioned at the start about possible links between malaria and COVID-19, and there are a couple of people who would like you to elaborate on that. So if I may.
Okay, excellent. Excellent question. Yeah, so the possible link, because of COVID-19 imposes this restraint, people have to stay indoors. Okay, so because of lockdowns and so on, people are not, so are staying indoors, which means they are becoming more vulnerable. Malaria mosquitoes bite at night, okay, so they are more exposed to malaria mosquitoes. That's what we're seeing. And also the people who have to go and do the indoor residual spraying and also distribute the bed nets will not be able to do so because they all have to stay home. So that's what I think needs to be quantified. What is the impact of not being able to do all those things on on malaria disease? And I think it's making it worse, definitely, because entomologists are not able now to go and do the spraying, particularly during the first 2 waves of the disease, because everybody stays home. So we're not spraying, and also the people who are going out there to distribute bed nets are not able to do so. And that's the point I was trying to make. So COVID-19 impacts many other diseases, not just malaria, I think. And another thing is clearly it impacts seasonal flu. This year, for example, data from the CDC say that seasonal flu burden is much, much lower than it's been in decades. And the reason is simply because some of the residual effects of the interventions that we are applying against COVID is also useful against seasonal flu. Okay, so we were masking up, and masking also helps us to not get seasonal flu. So there are all these connections interconnections and the co-infections, the impact of one disease on another? But that's a very good question, and I'm glad it's been asked.
Okay, and there is one question that came in the chat, and in answer to that question, yes, this lecture will be is being recorded and will be put up for everyone to see on the AMS YouTube channel. I'm not sure how long it will take for that to happen, but it will happen sometime in the reasonably near future. So people will have a chance to see it again, and those who didn't have the opportunity to see it live will have a chance to see it. Let me just ask, since we are really out of time, let me just ask one more question. I did have somebody who asked whether, and I think it's a good question, whether in addition to asked about confidence intervals for your models. So let me just,
sorry, can you repeat that? I didn't hear that.
Asked about confidence intervals for your models.
Excellent question. That's an excellent question. So again, models are formulated based on some underlying assumptions, right? You have to make assumptions in order for you to make models, to formulate models. So and also that the model contain parameters, so many parameters, right? So if you have parameters, there's always uncertainty in the estimate on some of the parameter values. You cannot know for sure each and every parameter value, each and every parameter, what precisely its value is. So that's underlying uncertainty in the estimate of the parameter. And that's why we have to do uncertainty analysis. These are statistical techniques like Latin hypercube sampling method, where each parameter is defined as a distribution, and you sample out of each of those. And then you look at, instead of just doing one realization, one simulation, you do a collection, like 1,000 different runs, and then you look at average rather than. So that's a great question. You can all, if you have 2-3 different people writing a model for a disease, the chances are that they all come with different models. So that's always uncertainty in model selection itself, but there's even more uncertainty in the actual model itself when you have many parameters in the parameter. So it's parameter uncertainty in the model selection, and also parameter in, like I mentioned, climate change in some of the models, climate aspects. So there's uncertainty in the climate models we use to predict the temperature as well. So all of these things together, and that the reason why this is so important is highlight the importance of statistics. Statistics plays a major role in what we do, because we have to, when you give predictions, of course, you have to give certain confidence intervals. So we calculate, when we do all the fitting, we have to calculate the 95% confidence. All of those things we have done in all of our models. But I want to highlight even the bigger picture, that when you do all of the things, there are lots of parameters, there are lots of sources of uncertainties. So uncertainty quantification is a new field now, thriving field in statistics. That is huge. That is machine learning, combined with machine learning. That is really, really important, and require a lot of mathematical theory and rigor behind it. It's a serious. So this is an important point, because we design models. If you want the models to be realistic, okay, the models typically have to be large in general. Although the general framework for me is I like to start with simple model, the simplest possible model, and then we'll see if it doesn't work, okay, what do I need to add to the model to make it work, to make reasonable predictions. But in general, realistic models are typically large and nonlinear and many parameters. Therefore, all these issues with uncertainty quantification come in many different ways. And how we quantify uncertainties is a serious science. I'm really glad this question was asked. But I want to highlight, particularly to the students here, please take a lot of statistics courses, probability theory courses, machine learning courses, because this is very useful in enabling us to design good models and using this model to make predictions, particularly if we're using models to make forecasts. Like the group in Washington State group, Chris Murray's group, I mean, they make the forecasting thing, right? Heavy duty statistics. And that's why the Presidential Task Force was using, each day they come in to do the media interview to state, okay, how many cases, the expected cases next. It's based on this serious machinery, statistical machinery. But anyway, again, it's the coming together of mathematics, probability theory, all branches of mathematics, analysis, algebra, geometry, all of this together, and then working together with statistics and computation. So I'm really glad the question is asked.
Okay, well, there are several more questions. I'm sure we could go on for hours with this very interesting topic, but we don't have hours to go on. I think we're going to have to bring this to a close. So thank you once again, Abba, for this really interesting talk. As I said, for those of you who want to look at it again, will be available on the AMS YouTube channel. And thank you again.
Thank you so much. I appreciate it. And thank you all for coming.